Right, hey, seventh graders. So today what we did was we went over our homework assignment first. So if we look at our homework assignment here, we needed to fill out our Punnett Square first before we started answering any of these questions. So up here is the only way we can really start doing this. And so up here it says, one of the traits that the pea plants of Mendel observed was seed shape. And the seeds were either round or they were wrinkled. Mendel observed that the round seed shape was big R, which means it's dominated. So round seed shape is dominant. And then it says wrinkled seed shape, little r. So if it's not dominant, if wrinkled is not dominant, then it must be recessive, right? So anytime we see a dominant allele, so right here we have a dominant allele, it will mask this recessive allele. This dominant allele of this other parent is dominant. It means it's round seed shape, so it will dominate the recessive one. So we know that this parent must be round seed shape because of the dominant allele. This parent right here is the same exact type as this one. And so we can write here, this big R comes down, this big R goes over. Here, let me, I'll write it sloppy. So this R comes down right here, big R comes down. Whoa, let's go back. I'm sorry, everybody. Long day. Make this thinner. Okay. So this R comes down. This R comes down again. This little R comes down. And this little R comes down. Now I come over to the side. This big R's passed over here. Big R's passed over here. This little R's passed over. This little R's passed over. Okay? So now we have our Punnett square that's filled out. And so let's remember, what's a heterozygous individual? So it does say something about being heterozygous and homozygous. So heterozygous. Heterozygous means different, right? So it means different alleles. So this guy right here has different alleles, big R, little r. This guy has big R, little r. So this is a these two would be heterozygous. Then we have homozygous. Well, we have two types of homozygous. We have homozygous dominant and we have homozygous recessive. Which one's this one? This one right here is homozygous recessive because it has little r. So our homozygous recessive, but then this guy's the same. So homozygous means the same, so it's the same alleles. So homozygous recessive, homozygous dominant up here because these alleles are dominant. They're the same. All right, and that's this guy right here. All right, so let's get on with this. So it says, what two forms of trait for seed shape did Mendel observe? He observed the wrinkled and round. It says up here. Man, last sentence. What was the phenotype for parent seed? So phenotype is physical, remember? Physical traits. So round, were they round or were they wrinkled? Well, the parent here, we already said, since he has the dominant round allele, and this guy has the dominant round allele, then they must be both round. What's the percentage of possible types of offspring that had the same genotype as the parent? So if you guys remember, the genotype is the allele combination or what we cannot see. So this big R, little r of the parent, that's a genotype. This big R, little r, that's a genotype. That's the combination of alleles. This is a combination of allele, big R, little r. This is an allele, or sorry, a genotype. Genotype. So it's saying what's the percentage of possible types of offspring that have the same genotype as the parent? So this parent here has big R, little r. This parent has big R, little r. So 1, 2, look the exact same as this parent. This guy looks like, so 50%, 2 out of 4, right? Number 4 says, what's the percentage of the possible types of offspring that are homozygous? So we already said this guy's homozygous and this guy's homozygous. So 2 out of 4, that's 50%. What percentage of possible types of offspring that had the same phenotype, so physical features? So which ones are going to be round like the parents? This guy's going to be round because he has the dominant allele for round. 2 dominant alleles for round, so this guy will be round. So that's 2 so far. This guy will be round with a recessive allele, but this will always mask the recessive allele. This guy will be wrinkled. So there's only 3 out of 4. 3 out of 4 is 75%. Number 6, what's the genotype? So it's only asking for a genotype here of the offspring that did not share the parent's phenotype. This is the guy that did not share the phenotype. Remember, he's wrinkled, so what's his genotype? It's little r, little r. And it says, what's the probability that 2 heterozygous parents, are these guys heterozygous? Yes, they are. They're different, right? Big R, little r, big R, little r. So we've already done this. So then it says right here, what is the probability that 2 heterozygous parents, like we have above, would have an offspring that produced round seeds, right? This guy's going to have round seeds we've already talked about. He's going to have round seeds, and he's going to have round seeds. So 3 out of 4. A ratio is comparison of numbers. So how many there is versus how many total. So 3 to 4 or 3 over 4 is how you write a ratio. And then what is genetics? Genetics is the study of traits passed down from parent to offspring. This number 9 says, what role do genes play? Genes control what we look like and how we act. What are 3 principles? I only required you guys to do 1. But traits are passed down from parent to offspring. That's what Gregor Mendel found out about pea plants. And then traits can be dominant or recessive, meaning one trait you get from one parent, maybe different from another trait you get from the other parent, but only one's going to show in front of the other. So whichever one shows over top the other is the dominant allele or the dominant trait. Okay, so moving on. Today we did this lab here. We also took notes. So you guys, I would do notes before you do this lab here. So when we did this lab, we needed 2 bags. I didn't have paper bags, I had plastic bags. So here's bag 1. All right, here we go. Let's see if I can view, zoom. All right, so we can read this a little better. So could you predict how brown rabbits would result from crossing 2 heterozygous black rabbits? So heterozygous means different, right? You can see it right here. This is a heterozygous guy, big R, little r. So heterozygous means one's going to be capital letter, the other allele in that offspring or in that individual will be the lowercase, right? And so we have 2 bags. Here's one bag, and here's my other bag. So each bag represents a parent. And so in here, I have 2 sets of DNA. Well, guess what? How many sets of DNA do we all have? We all have 2 sets of DNA. As a child, though, or as an offspring, how do you get those 2 sets? Well, you get, because of meiosis and sexual reproduction, you get one set from one parent and the other set from the other parent. Now we see how this one's colored, and this one is this plain Jane regular tan popsicle stick. This is a painted popsicle stick, okay? And so what's going on here is we're trying to see how we're actually working out a Punnett square is what we're doing. Like we're physically doing it by drawing these out. And so when we look at a percent is actually looking out of 100. So if you do 70%, that's 70 out of 100. If you're doing 15%, that's 15 out of 100. That's essentially how it's working anyways. Okay, so we have our two bags. We have 100 tan sticks. So I have 100 tan sticks. I have 50 of those tan sticks in here. I have the other 50 in here. And then it says 100 colored sticks. Well, I have 50 of those colored sticks in here. And so then I have 50 colored sticks in here. So I have a mixture, right? Well, it says, you guys look here, it says 2 heterozygous black rabbits. So heterozygous, that means their alleles, they have 2 alleles, right? But one's dominant, one's recessive. Well, in this activity, this came from the same bag. We have a dominant and we have recessive. So this is what the parent looks like. It has the dominant allele, which is colored stick, and we have the recessive allele, which is the tan stick. How do we know this? So down here, we start looking at this. Number 1 says, use a Punnett square to predict how many color-to-color combinations. So what that means is if I draw a color out of one parent, one bag, right? And if I draw a colored one out of the other parent bag, how often is that going to happen? Well, we can use a Punnett square to predict that. We've done it already. So that's color. And then if I do this again, what's the probability of me next time pulling out color tan? So color from one parent and the tan stick from the other parent. What's the probability of that? That's what the Punnett square is for. And then finally, we get to the tan, which is the recessive. One from one parent, the other from the other parent. Again, we're predicting that right away. And then how do we know what's dominant? How do we know what's recessive? Well, it says right here, colored sticks represent the black alleles. We know black is dominant because up here it says crossing 2 heterozygous black rabbits. So if they're heterozygous, right, if we peer right through here, this big R is this big R, little r guy right here. Like this is an individual, right? That's their genotype. We know that this will always mask the recessive. And so if black is, if an organism is heterozygous and they're displaying a trait, that trait must be dominant. And so black is dominant here. Therefore, the colored sticks represent the black alleles, and the tan sticks represent the brown alleles. Brown must be then recessive. Okay? And so when we go to, you're going to go ahead and draw that here. And this is heterozygous, remember? And if we look right here on our homework, guys, look right here. Heterozygous parent, you're going to draw the same one. So it's going to be, right, 25% will be homozygous dominant, 25% should be homozygous recessive. So what's that saying? We're betting that, remember, homozygous dominant would be color. Homozygous. So this means if we do 100 draws, we should do this 25 times, right? 25% or 25 out of 100 is the same thing. Now it's saying if we look, that was this guy, that's homozygous dominant. Now if we look here to heterozygous, which would be color tan, right? That would mean if we're predicting how many times will we pull out a colored tan from one parent and a colored from the other parent. How often would that happen? That should happen, well, according to our Punnett square here, dominant recessive, that should happen 50% of the time. So that means we should pull this out 50 times out of 100. And then finally we get our recessive. How often are we going to pull out one tan from one parent and then another tan from another parent? And right here it says in our Punnett square, 25% of the time that will happen, or we're predicting that will happen 25% of the time. Okay? And so I'll go ahead and give you the numbers for this. Okay. So anyways, like I was saying earlier, what I'm doing here, my setup, I have one bag here. This represents the male bag, and this one represents the female bag, and I randomly draw out of these guys. Here's one allele for them. So every time I do this, I draw this out. This is one trial run. That's one trial run. So I'm going to jump back here, and I'm going to mark on here what I get. I got color. Okay, so this is what we did in class, by the way. So I'm going to mark X. Okay, that was our first one. Now I'm going to do it again. Randomly, I'm not going to look. Oh, I got color again, right? So we're predicting that this color, if you remember, this is going to happen 25% of the time. Now, will it actually happen 25% of the time? Probably not. But overall, we would think it would happen 25% of the time. Okay? So what this whole lab is about is you or us, we're doing this actually, and then we're testing it against our Punnett square that does the prediction and see if the Punnett square actually comes out somewhat accurately, right? And so let me go ahead and tell you what your numbers are. Let's go ahead and make this guy 30. So color to color. Let's say I drew color out. We did that 30 times out of 100. And you can see obviously right here that there's 100 trials. Okay, so let's say 30 for 30 for color tan, let's say 48. And so then these are numbers I'm just pulling out of my head, so I need to write these down. So then if this is out of 100 draws, right, then I just add up my 30 plus 48, and then I'm going to subtract that from 100, which gives me 22. So then that means I drew tan 22 times. So I draw out of the 100 times that we did our drawings, I drew tan 22 times. So anyways, what that means now is you got all the information you need for this. So out of those guys, you look back here, you're going to look at these three numbers right here the entire time you're doing 1 through 4. That's all you have to do is questions 1 through 4. Make sure you write down this Punnett square. So that Punnett square I was showing you earlier about the heterozygous black rabbits, make sure you cross those. So which combination occurred most often? Back here. So did color occur more often? Did color tan? Did tan occur more often? So then you write these down. What was your ratio? So then our ratio would be how many times did I actually draw out color out of 100? So how many times did I draw color? That's X. That's for you to figure out. It shouldn't be too hard. Like I said, it's the numbers right here. You're going to do that for color tan. What's going to be the hair color of these three different things? Now you're going to compare your predicted or expected results. So this means your Punnett square that you're drawing up here, this was your predicted or expected results. how did this go along with these guys that you actually drew out? So all you're saying, you don't have to explain anything. You just have to say, we predicted 25% would be color, but we actually got 48% that were color. That's all you have to say. We predicted color tan would be 50%, but actually it was 30%, something like that. That's totally fine. You can write this down here, fill this in, and then hypothesize. So you didn't get your 25, 50, 25 like we predicted on our Punnett square. How could we get better predicted results that are closer, that would make get closer to the actual results? Or how would we get our actual results closer to our predicted results? And so you'll turn that in for me. All right. So if you got any questions with that, let me know. Bye.